Hi, science friends. This is a review not from yesterday, but rather flashing us back to biochemistry because we have to keep reviewing this stuff. And that was a hard unit for you guys. So let's discuss the four macromolecules and their monomers. You all should really know this. I don't know why this is something that's so hard for students every year, but you've really got to nail these macromolecules and their monomers. I think that was actually one of the questions on the test in the last unit on the free response. You had to name macromolecules in the cell membrane. And some of you all couldn't name, you named things that weren't even four of the macromolecules. So this is going to come back. It was on the cell test. You'll see these things again. And so it's important that we keep revisiting them. Macromolecules, or sometimes they're referred to as biomolecules. So you should tell me proteins, carbs, lipids, and nucleic acids. Okay, so proteins, remember proteins are made up of amino acids. That's the monomer. Amino acids have the group, they have the carboxyl, the amino group, and the carboxyl group, and then an R group on them that makes them different. Okay. They are for, we've talked about what a lot of things for proteins are for. They are messengers in the cell. They help with structural things. They help with transport in facilitated diffusion and active transport. They form protein channels. Proteins can be hormones. We can have lots of different things that proteins are important. So that's proteins. Let's look at the next one. Carbs. The monomer of carbs are simple sugars. Remember, carbs or simple sugars are always going to end in ose. That's how you recognize a sugar. They are going to be made up of CH2O. So there's always going to be twice as many H's as there are C's and O's. For example, glucose is C6H12O6. So there's twice as many H's as there are O's. Okay. Lipids. Lipids are going to be made up of a glycerol and a fatty acid group, or fatty acid side chains. So lipids are important in the phospholipid bilayer in the cell membrane. Some hormones are also lipid-based. You can have hormones that are proteins and hormones that are lipids. Remember, just to review, hormones are long-distance signaling. They travel through the blood. It takes a little bit longer for them to respond, but they have a longer duration. And they would bind to like a signaling. They could be like a ligand that would bind to something and cause a signal. And then the last is a nucleic acid. So nucleic acids, an example would be like DNA and RNA. And so they are made up of monomers called nucleotides, which have three parts: the 5-carbon sugar, the phosphate group, and the nitrogen base. Okay, so remember your 5-carbon sugar, that's going to be deoxyribose in DNA, ribose in RNA. The phosphate group is just it's just a phosphate group. Those make up the backbone of the DNA molecule, or the sides. And in the middle, we have those nitrogen bases, which are A, T, G, and C. And they pair according to Chargaff's rules. A pairs with T, G pairs with C, and we'll revisit that a lot more later. Okay, so that's just to review the four macromolecules and their monomers. This stuff is so important, guys. So important. All right, so we are going to talk about enzymes today. And enzymes fall under the class of macromolecules as proteins. So most enzymes are proteins, okay? And while we say most, really when I think about it, I think of all. So for the most part, if I'm talking about an enzyme with you, we are going to call it a protein, okay? AP doesn't expect you to be able to identify any that are not proteins or anything like that. So enzymes, you will notice, you may want to make a note of this, end in ase. Okay, so we said sugars end in ose, enzymes end in ase. And what enzymes are is they are a biological catalyst. So they're found in living things, and they are a catalyst. So what a catalyst does is it helps to speed up or get a reaction started. Okay? So reactions can happen, chemical reactions can happen over long periods of time by themselves. But if we want to speed them up, then we can add a catalyst, or in a living thing, a biological catalyst, because it's a protein that's found in a living thing, is an enzyme. Okay? In chemistry, we don't use enzymes because generally we're studying chemicals in a test tube. We're looking at chemical reactions outside of the body for the most part. But in biology, because we're looking at chemical reactions and metabolic functions in living things, then in living systems, these catalysts are called enzymes. So that's kind of the difference there. Enzymes are a specific type of catalyst found in living things. So let me give you an example of a chemical reaction with something that you may know. Now, this isn't going to involve an enzyme because it's not a living thing. But let's talk about hydrogen peroxide for a second. You'll probably all know what hydrogen peroxide is. It comes in the brown bottle. If you have a really bad cut or scrape or something and you need to clean it out, you may pour hydrogen peroxide on it. And it'll bubble and fizz up a little bit, and it kind of stinks, and it kind of kills all the germs is what its function is. And so they store it in the brown bottle because it's light sensitive. And if light, if it's exposed to light, then that can sometimes cause it to break down. Well, when hydrogen peroxide breaks down, it breaks down into water and oxygen gas, okay? Water and oxygen gas, and that chemical equation is not balanced, but you could go in and balance it. Water and oxygen gas. And so if you look at a bottle of hydrogen peroxide, it has an expiration date on it, okay? If you let it go past the expiration date, and sometimes honestly even before the expiration date, then if you poured it on a bad cut, it probably is not going to fizz and bubble up. And the reason is because the hydrogen peroxide has broken down into water and oxygen. So you're not putting hydrogen peroxide on your cut. Instead, you're just pouring water on the cut. And that's not going to cause it to fizz and bubble. Okay? So this reaction will happen, the peroxide converting to water and oxygen will happen on its own, but that takes like 3-ish years for it to occur. And that's why the expiration date on peroxide is usually pretty far out, maybe even longer than 3 years. But you could add a catalyst to this chemical reaction. You could add, it's not an enzyme because it's not in a living thing. If you add manganese oxide to this chemical reaction, it'll break down hydrogen peroxide to water and oxygen like immediately. Okay, and so that's a catalyst. It causes this reaction to happen faster. Okay, naturally it'll happen, but it'll take a long time. But if you add this catalyst, it'll do it immediately. Okay, it speeds up the chemical reaction. And so that's what an enzyme or a catalyst does. It speeds up a chemical reaction to make it happen faster. Okay, so what enzymes act on is referred to as the substrate, okay? So manganese oxide in this case is our catalyst. Again, it's not an enzyme because it's not a biological system, but what it acts on is the substrate. So in that example, the hydrogen peroxide would be the substrate, okay? Here in this diagram, our enzyme is sucrase, and what it's acting on, the substrate is sucrose, okay? Notice those similarities in the names. The enzyme and the substrate that it acts on, not always, but often mimic one another, okay? Sucrase is the enzyme that helps break down sucrose into glucose and fructose. So sucrose here is a carbohydrate. We know that, going back to our macromolecules, because it ends in ose, okay? And it breaks down into 2 monomers called glucose and fructose, 2 simple sugars, okay? So this is a complex sugar with more than one simple sugar, and then it's going to break it down. Okay, so the enzyme is here. What it reacts on, or what it acts on, is usually on the reactants, or always rather going to be on the reactant side of the equation, on the left side of the equation. All right, so when we add an enzyme, it's what's known as the activation energy. That's the energy that's required to get a reaction started. Okay, so if we add an enzyme, this activation energy is less. Okay, so here's the better diagram. Here's the activation energy with an enzyme. Here's the activation energy without an enzyme. So you can see it lowers the activation energy. What this yellow line here shows is the amount of energy that's released. So it doesn't affect the amount of energy that's produced, and that's important. The reaction still proceeds the same way, but it just lowers the amount of energy it takes to get it started. Okay, so just like signaling molecules fit in a protein, a receptor protein, in a very specific, precise way, enzymes and substrates also work the same way. They're very specific to one another. And when you pair an enzyme and a substrate up together, it makes what's called an enzyme-substrate complex. Okay? Now, another thing, I don't think I have written notes on this, that you may want to jot down about an enzyme, is enzymes are not used up in a reaction. Okay? They don't participate in a reaction. So when we see it here, we don't see it as a reactant. You all know what reactants and products are from chemistry, from biochem last year. It's on the arrow. It's not it's recycled. Enzymes can be recycled. They can work over and over and over again. Okay? Now, they may all be used up kind of for like a finite time period, kind of like ATP. We saw yesterday how it cycles back and forth to ADP and then gets that phosphate back on it. So all of the enzymes can be occupied at one moment. They can all be busy doing a job. But after they convert that sucrose into glucose and fructose, like in this example, then sucrase is ready to do it again. Okay? It's going to bind to sucrose and do its thing, but after it does its thing, it's done, okay? And it can be reused. It can be recycled. So they're not used up in a chemical reaction. They don't participate in a chemical reaction. They're not a reactant, and so that's why we write them above the arrow, okay? So here we have, on this side of the equation, we have our substrate with our enzyme, okay? And then our enzyme substrate complex is kind of formed there in the middle. And then over here on the other side, we have our product, and our enzyme is still present. So you see that the enzyme is still there. It doesn't go away. It's not used up. Okay. So the last thing I want to talk about is this active site. Okay. So just like I mentioned a moment ago with the signaling molecule or the ligand binding on the receptor protein in cell communication pathways, the enzyme and the substrate comes together and bind on in an area called the active site. Okay, so here's our substrates. Oftentimes in a chemical reaction, there's more than one. Okay, so the substrate here is sucrose plus water. And so those two substrates come together and they fit perfectly in this active site on the enzyme. Sometimes it's referred to as an induced fit. Okay, so the enzyme changes shapes to mold around it. Okay, the enzyme kind of absorbs it and, kind of forms like clay around it. Okay, and then the substrate, you see in this picture, we start with a red and green substrate. They get converted to products. So we have something different. We started with sucrose and water, and we were left with glucose and fructose. So here's our sucrose and our water, and we're left with glucose and fructose. Okay, so we get our products that are released, and we have our enzyme that's still available to be reused. Okay, so here in this stage, it's being utilized, but once those once those substrates are converted, it's available to be reused. Okay, for that enzyme and substrate to come together in the enzyme substrate complex at the active site. Okay, the active site is where the action happens. The active site on the enzyme, that's important. We got to know about active site. Okay, we also have to know about induced fit. Both of those are very important. It's not exactly like a lock and key fit. Sometimes you hear the term lock and key fit, and this is not a lock and key fit, because when I stick my key in my lock, the lock doesn't mold to my key, right? Here, the enzyme molds to the enzyme substrate complex, and so that is referred to as an induced fit. Okay. All right, so that wraps this up today with enzymes. We'll do a little bit more with enzymes in our next lesson, and you all have a great day.